Very welcome to today's Global Sepsis Lecture. The STAIR Sepsis Lecture online today is going to be by Associate Professor Dr. Tafese Tufa from Ethiopia. Tafese works at Arsi University in Asella in Ethiopia. And he's also the co-director of the Hirsch Institute of Tropical Medicine, which is a collaborative project between Arsi University and Düsseldorf University, Heinrich Heine University here in Düsseldorf in Germany. Very welcome, Tafese. Thank you for doing this. You are going to talk about novel diagnostics in sepsis-causing pathogens today. And I'm very much looking forward to your expert view on the topic. Whenever you're ready, I'm happy for you to start.
Okay, thank you, Ruth. Good afternoon, good morning, everybody. My name is Tafese from Arsi University. Then I'm so happy to talk about novel diagnostic for sepsis-causing pathogens, because we know that sepsis is contributing for high mortality. One reason is since we don't have definitive diagnosis for this disease. So I'm happy to present what I have found from literature and also including my experience. To present this lecture, I don't have any conflict of interest. Here, these are my objectives for this presentation. So I hope at the end of this presentation, everybody should understand the advantage and the disadvantage of current sepsis diagnosis method we have, particularly about blood culture. Then also explain the role of liquid biopsy in detecting sepsis and antimicrobial resistance genes. Liquid biopsy means just cell-free DNA. And recognize the advantage of integrating host microbes, plasma metagenomic for sepsis diagnosis. This is something novel, recently, tried in a different country, and also some literature support this. Identify the future of an ideal diagnostic. So I'm happy to start with a question, just which says, which method is the most sensitive and specific for rule out in sepsis? 1, blood culture. 2, multiplex PCR or PCR. 3, integrated host microbial metagenomic next generation sequencing. 4, cell-free DNA metagenomic next generation sequencing. I understand most of you can answer, but I love if we answer at the end of this presentation together, because I don't want miss. Just from all participants should answer this. That's why just at the end of this presentation, we'll answer together. As an introduction, this is what I found from Global Sepsis Alliance. As you know, annually up to 50 million sepsis cases can be happen. Then of these, about 11 million died. That means 1 day every 2.8 seconds. And also it contributes up to 20% disease worldwide is associated with sepsis. Not only days, those survived also, they may develop lifelong disability or organ damage. As a definition, we know sepsis is life-threatening organ dysfunction from a dysregulated host response to infection. Infection because of bacteria, can be virus, can be protozoa, can be also germ. So early diagnosis and identification of these underlying microbial pathogens is essential, because this is important for timely and appropriate antibiotic therapy. which is critical for survival of the patient. So far as evidence shows, only about 30% of the cases, about 30% of cases, no etiological pathogens is identified. That means we need definitive appropriate diagnosis for sepsis. As a result of this, antibiotic treatment often remains empirical rather than pathogen targeted. That means we don't know the cause, and also we don't know which drugs will work, but to save the life, clinicians start with broad spectrum antibiotics. So most of the clinical decision made based on epidemiological information, the data they have from locally or nationally or internationally, than individual patient based. This decision may, lead to overuse of antibiotics or misuse of antibiotics, which also have a consequence because one, it can, when we use these broad spectrum antibiotics, MDR bacteria, multi-drug resistant bacteria, their population can be increased, and then so to give the chance for emerging of drug resistance. Also, like Clostridium difficile, say those antibiotic-associated hospital-acquired or opportunistic infection can be happened. The challenges with sepsis diagnosis with existing, with what we have, blood culture, as I said, it only covers a small portion of the pathogens. It also, takes longer time to provide the results, and the pathogens remain undetected. More than 50% or even up to 70% of those possible cause sepsis we may not cover by using blood culture. The screening tools we have also for sepsis like QSOFA, Square, SEERS, and other criteria that also lack accuracy. They are not specific as well as not sensitive. So when we go to the treatment, the treatment decision will be empirical. That means, especially in the resistance era, our empirical try just will be inappropriate, about 50% or more than 50%, which leads also to expand the antimicrobial resistance and also become costly and just increase the hospital stay. Not only cost resource, it may be also cost life. That's why we have high mortality rate due to sepsis. We did some study 3-4 years ago in Ethiopia. What we did is we included into this study all febrile hospitalized patients, and we offer for all of them, blood culture and the blood smear, also some point of care test we have. However, we only found 5.4% blood culture positivity rates, which is really very low. You can imagine we provide standard blood culture, which is 2 sets, both anaerobic, aerobic bottle, general 4 bottle, but what we found is only for 5.4%. So we don't know about the rest 95%, even if they suspected for infection and hospitalized the febrile patient. Among those, what we found is just 38%, 38 gram negative bacteria and 26 gram positive bacteria. Among isolated gram negative bacteria also, more than 82% of them SPLS producing bacteria. We also try to check the empirical initiated antibiotics specifically for gram negative bacteria, and we found that more than 72% of those initiated likely ineffective when we compare with ST result we found. So this clearly shows that the blood culture we are using is not covering. We didn't stop here, and we also just transport negative samples, the plasma, to Germany, and we used multiplex PCR, just targeting to the common non-culturable pathogens like Plasmodium species, Borrelia, Rickettsia, and the leptospirosis, and arboviruses. This data also published. And what we found, almost half of them, the cause of febrile, like non-cultural, like Rickettsia, Borrelia, the Plasmodium. So we could increase, by using this multiplex PCR, but still it's not much. So this clearly shows we need further definitive, universal, and wide range, which can cover a wide range of pathogens to diagnose sepsis. Then when I came to the cell-free DNA or liquid biopsy, this is now which we are planning within states also to use to diagnose sepsis. What is cell-free DNA? The cell-free DNA is non-cellular DNA contained within a biological body fluids, can be like plasma, urine, CSF, and or other clinical specimens. So when we centrifuge blood, we can find also the cell-free DNA in the plasma. The source, this cell-free DNA can be, from circulating tumor cells or from the cell-free fetal DNA and the donor-derived cell-free DNA, and it can be also microbial cell-free DNA, which we really interested. The history of this cell-free DNA, it was for the first time reported in 1948, the presence of this cell-free DNA in the era of cancer patients. Then the concentration is different from patient to patient. That means it is high, for example, lupus patient, this cell-free DNA. And the idea of this cell-free DNA as biomarker initiated in 1966. You can see from this picture recently, this cell-free DNA, actually it's not almost a decade, used as a biomarker to diagnose sepsis. Before that, in 2000, this cell-free DNA was used as to check the prenatal abnormality by using this cell-free DNA testing from plasma. The origin of this cell-free DNA also from different tissues or different cells. As the study shows, for example, comprehensive human cell type mutilation atlas reveals origin of this circulating cell-free DNA in health and disease sepsis patient. You can see from this pie chart about 32%. The source of this cell-free DNA is from granulocytes, that is nucleated white blood cells, and also following that erythrocytes, progenocytes about 30%, and 12% in lymphocytes. But this will be changed when the patient, or when we come to the patient, or if there is a sepsis. As for instance, here we can see sepsis patient where liver organ is involved or damaged. You can see the origin of the cell-free DNA is from hepatocytes cells, the red one shows. And this also, go with alanine aminotransfer or ALT enzymes or liver enzymes, just directly proportional. When the concentration of the cell-free DNA of hepatocytes increased, the enzyme secretion also increasing. The same for sepsis, sepsis patient included into this study 031 code. As you see, this is bloodstream infection. The source of this cell-free DNA is from granulocytes. So when we have this cell-free DNA in the plasma, it also clearly shows us which organ was damaged. When we compare this cell-free DNA from microbes and from human, there is a slight difference. The first difference is the size, their size. The cell-free DNA from microbes is shorter, just up to 30 to 10 base pair. but the average from human is about 167. In the packing, the package also, there is histone here, but in the microbes, there is no histone. And the size distribution, usually if it is from microbes during sepsis, it is irregular. That means when the sepsis is there, it is broad and then increase, and then immediately it's gone down. Stability, as you can see from the table, it is really very a little bit stable in human when it is from human cell-free DNA, but the cell-free DNA from microbes is because of the sensitive to nucleus, it is not stable. So we should immediately separate it from the blood, the plasma, to extract this cell-free DNA of the microbes. The half-life from in case of cell-free DNA from human, it is 15 minutes to 2.4 hours, but it is less than an hour in total if it is from microbes. But as far as the microbes is there, still the shedding from these microbes is continuing. The use those cell-free DNA from human we can use to screen cancers, prenatal and transparent, but these microbes can be used for diagnosis, the presence or absence of infection. In short, the microbial cell-free DNA fragmentation are generally shorter, less structured, and more degraded than one cell-free DNA. The diagnostic of this cell-free DNA by using metagenomic approach, we can use this for different purposes, for different infections, like bloodstream infection, TB, fungal infection, and any complicated pneumonia. But the sensitivity is not the same. When it is the bloodstream infection, it can be higher, but when the infection is, or the source of the sepsis is prepared, the concentration may be low. This is how we can, get this cell-free DNA from this figure. You can see if there is infection, the cell-free DNA released to the plasma from tissue or from the microbes, because the microbes will be killed by the immunity activation. And also when you use antibiotics, they may release to the plasma. And if we just withdraw or draw blood and the centrifuge, we can get the cell-free DNA in the plasma, and then we can prepare library and then sequence. Then we can get the presence of which type of bacteria or virus, because this is only for DNA. You can also detect the DNA virus. Then with the combination of the medical history, we can use to manage our patient. The total concentration of this cell-free DNA also has, clinical implication. This means if there is infection or sepsis, there will be tissue damage or tissue injury, and the microbes release this cell-free DNA to the plasma, as we discussed. Then when we measure the mass of this cell-free DNA, if it is high, to give us information that really that patient is at risk. So usually it is high among those non-survivor or poor prognosis of the patient. If it is low, just or moderate, the patient outcome will be better. And also, it is usually low among those survivors. This means this also work for non-infectious cases, So when we measure this total cell-free DNA, we can get, information, because the half-life of this cell-free DNA is very short. That's why it can tell us the status of our patient. There was a study done and published recently. You can see from this figure, they used blood culture and the metagenomic next generation sequencing of cell-free DNA from different clinical specimens, like blood, sputum, urine, and pleural fluids. As you can see, for example, from the blood, by using this metagenomic next generation sequencing, they found 57. from the blood. But when they use blood culture, they only found 22 positive. So the 91 negative. Here only 56 is negative. Just when we see the total, by using this metagenomic next generation sequencing of cell-free DNA, they found 147 positive, but by using blood culture, only 84 positive. So when we calculate the sensitivity, the total positive rate of this metagenomic next generation sequencing of cell-free DNA about 71%, but the blood culture is about 41%. And the other here, why it happened, when we see this, the metagenomic cell-free DNA for infectious disease. For example, you can see this metagenomic cell-free DNA sequencing result here. Human cytomegalovirus is covered with a pound 11. We know that this we can't culture. But here also from bacteria, Haemophilus parainfluenza, by using it was not detected, but by using this sequencing cell-free DNA found about 12, and enterococcus also. Even if we could culture it, and we can detect by using culture, they found 13 by using cell-free DNA sequencing, but they only found 2 by and also Streptococcus pneumoniae. Know that Streptococcus pneumoniae also one of the cause of sepsis. In this study, they found 16 Streptococcus pneumoniae by sequencing the cell-free DNA, but there is no positive by using Streptococcus by using culture. This clearly shows us that we need something very sensitive which can cover pathogens. When we compare this among those survived and the non-survived groups, you can see the red one is the dead and the green one is survived. You can see here the most isolated pathogens from the non-survived by using this metagenomic cell-free DNA sequencing, like the leading one is Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, coli, as well as Pneumocystis jirovecii were the most prevalent pathogen isolated among those dead or non-survival group. We know that, for example, we can't detect Pneumocystis jirovecii by using blood culture, but when we use such things, we can cover it, or we can detect that. The other recently published, the paper published last month, just detecting the pathogen microorganisms using this metagenomic next generation sequencing for patients suspected infection presenting to the emergency department. They include 170 patients, those who have both blood culture and also metagenomic sequencing result. Then what they found, the positivity rate is just completely different. 49% by using this metagenomics sequencing of cell-free DNA, but only 60%, 0.5 or 70% by using blood culture. So this is done in Germany, in the Sendo company found in Duisburg. So the quality, there is no doubt about the quality. Among these next generation sequencing positive cases, 63 had negative blood culture. Why? Because for instance, by using blood culture, they couldn't detect a single anaerobic bacteria. But by using this next generation sequencing, they detect 75 anaerobics from this suspected patient. So when we see this, we need something which covers all pathogens. I know also, as experience from our setup, in the last 5-7 years, we couldn't detect a single anaerobic bacteria by using plate culture, even if we use anaerobic bacteria also. As I said, the sensitivity is very high by using metagenomic next generation sequencing of cell-free DNA of the microbes from the plasma, but there is really a limitation. The first limitation is we can't easily distinguish infection from the colonization or contamination, because we are also full of bacteria as a commensalism or as a normal flora. There is high possibility that the DNA of these bacteria can be also found in the plasma. The other is difficulty of differentiating anemia from the true blood infection. Sometimes when we do this sequencing, we may find 3-4 microorganisms. Can be virus, DNA virus, or bacteria. The other, when we use this cell-free DNA, we can't also detect RNA virus-caused infection. The turnaround time and the workflow complexity also, because for this next generation sequencing, we can't do it for a single patient. We have to pull the samples together and run together, because if we go for a single patient, it may be not cost effective because it is expensive, and also the setup is, at least for maybe just depending on the type, 96 patient or 10 patient, or just it depends on the type of the next generation sequencing we use. The other one is the high cost or limited availability. The cost also very expensive, and it needs also expert, and you can't do such tests at every health facility. The other very important is interpretation challenges. We should have algorithm for this, how the clinicians use the results from this cell-free DNA sequencing, because DNA cannot separate live bacteria and dead bacteria, so it needs algorithm. For instance, one of the limitations, you can see the cost of this cell-free DNA sequencing or molecular tests for sepsis diagnosis. This is a molecular platform used in sepsis diagnosis as example. For example, here, if we use multiplex PCR real time for one patient, it maybe needs up to 6 hours and up to 20 USD. But if we go for multiplex real time PCR, it only for those targeted primer. If it is 9 for 9, if it is 6, only 6 pathogens, it cannot go beyond that. But within our project, we are planning to use export nanotechnology mini, which can be for 1 to 6 samples, and also can be faster, up to 9 within 1 day, or 7 to 9 hours, which cost also about 130. This cost is only the cost of the consumable and the sample. It will not cover the cost of manpower and infrastructure and things. So when that is included, still the cost is very high. So the next topic is integrating host microbes, plasma metagenomic sequencing, because this is very important. We have said that the plate culture has limitation, both less sensitive and also less specific, and the metagenomic cell-free DNA sequencing, highly sensitive, but still lack specificity. So to address this, the clinical impact of plasma DNA metagenomic has been, since its question, we should go for whole blood transcriptional profile offers the potential to get this limitation, because this cell-free DNA cannot, differentiate microbes in a certain clinical relevance. inability to detect RNA virus and the limited ability to rule out the presence of the infection. A sepsis diagnostic assay which combines this host transcriptional, that means host profile or gene expression, with just sequencing both RNA and DNA, that may be more accurate diagnosis to rule out sepsis. There was a paper recently, actually it was in 2022 published. It is just in the Nature Microbiology. It says integrated post-microplasma metagenomic for sepsis diagnosis in prospective cohort of critical ill disease. The researchers, what they did, patient from emergency department, those need ICU, they recruit into this study. And then they collect whole blood, they collect plasma, and from the whole blood, they also just check the host response or gene expression from the host. And from the plasma, they also consider RNA, DNA, and then also gene expression. They group the study participant into different groups, like as a sepsis, bloodstream infection. If the blood culture is positive, if they found any growth from blood culture, they group as sepsis, bloodstream infection. If others clinical sample positive for microbiology test, they group as sepsis, non-bloodstream infection. That means can be sputum or urine. If they found positive, they group as sepsis, non-bloodstream infection. If there is no growth, but suspected for sepsis clinically, they group as sepsis suspected group. And if there is no evidence of infection, but admitted at ICU, can be cardiac arrest or other things, they group as non-sepsis. Then here, what they found, for example, the microplasma DNA mass difference between the sepsis groups. This means the concentration of the cell-free DNA they found, it was high, among sepsis bloodstream infection, and followed with sepsis non-bloodstream infection, but less among non-sepsis patients. Not only that, what they found by blood culture and what they found with metagenomic generation sequencing, and because here they also consider within the metagenomic generation sequencing viral and also host or gene expression of the host. But still, they couldn't cover all pathogens those they found by culture. When we come to the peripheral site infection, that means sepsis, but those blood culture is negative, but other clinical specimen is positive. By using this metagenomic next generation sequencing, they couldn't cover all of them. For example, in case of lower respiratory tract infection, they only found 8%. But for urine, just 38%. That means there is high possibility still to miss or not to detect the presence of this cell-free DNA in the plasma. But if they combine, or when they combine together, they got that it's really sepsis. I will show you to the next slide. So the sensitivity of this blood culture as a reference standard was 83 and varied by pathogens, because some pathogens still couldn't be detected by using this metagenomic sequencing as well as the host expression. Pathogens were detected in 10 of 37, that means among those non-sepsis groups. This also clearly showed that there is high possibility that especially enteric bacteria or normal flora of GI can be detected in the plasma, even if the patient is for not develop sepsis. This integrated host microbial plasma metagenomics for sepsis bloodstream infection, as you can see from this figure, just only 2 viral additionally detected by using this approach. That means host microbes plasma metagenomic sequencing. Almost the others, when they measure the concentration of this cell-free DNA to consider as a positive, the concentration of this or the mass of this microbe cell-free DNA should be above 20 pp, as you see. So when this, it is almost so detected by blood culture, the concentration of this microbial cell-free DNA was above that. For host criteria, host sepsis classification, probability should be above 0.2 or 50%. When we see among sepsis, non-bloodstream infection, you can see the more here viral, the viral possibility is high compared to the bloodstream one. So in general, they detect 42 or 40 to 100% case of sepsis bloodstream groups by using this integrated host microbial plasma metagenomic sequencing. And also they could detect about 97 case of the sepsis from non-bloodstream groups, but they are suspected for sepsis for an overall sensitivity about 99%. When we come to the non-sepsis group, still the specificity was 78 among these non-sepsis groups, but some still detected, But those detected, when we see they are enterobacterial family, from GI, those normal flora, they show that this patient is non-sepsis group clinically and also by using machine learning, but still there is possibility of the microbe cell-free DNA in the plasma. That's why if we only focus on molecular method, it did not definitely tell us that it's really sepsis due to these microbes or not. That's why if we combine the gene expression of the host, we can rule out almost 100%. Within the no sepsis group, about 27 patients had a pathogen detected by this next generation sequencing. But as I said, almost 90% of them, they are normal flora of the GI. So it is possible that sequencing were derived from these non-viable organisms unable to grow in culture. The detection of these pathogens alone was in many cases insufficient for sepsis diagnosis, but when combined with this host transcriptional profile, we can easily rule out the sepsis. So when I come to the answer of the question, as we promised, now the answer you can put in the chat. The question is, which method is the most sensitive and specific for ruling out the sepsis? Blood culture, multiplex PCR, or integrated host microbial messengers next generation sequencing for cell-free DNA metagenomic next generation sequencing.
Okay, I am looking at the chat and I see a lot of threes. So there is Gary, Dejene, Cody to Ethan, Gaby, you're all saying 3. Is that right, Tafese? Answer number 3. Is that the right answer? That's the majority of people in the chat.
Yes, the answer is 3. Reason, the blood culture, we know that still it is gold standard, but it's less sensitive and less specific. Because sometimes there will be also contamination like cons and other things. PCR, multiplex PCR, as I try to mention, it only, the primary you use. it only target the primary use. So it means they're sensitive to loud sepsis, not to cover all pathogens. The cell-free DNA metagenomic next generation sequencing, the problem is it is highly sensitive, as we discussed, but it also less specific. It lacks specificity because there is high possibility that we can detect the cell-free DNA in the plasma of the patient as commensalism or contaminant of during sampling. But if we integrate host microbials, next generation sequencing, because we also, when we found these microbes in the plasma, we also simultaneously, when we check the gene expression or signature to the certain specific infection, can be viral, can be bacteria, then we can rule out that it is contamination or really it is a cause of sepsis and pathogen. So while the cell-free DNA metagenomic next generation sequencing is very sensitive, the targeted host microbials provide the best balance of sensitive and specific in sepsis diagnosis to rule out. So it is right. The answer is 3, integrating host microbial metagenomic next generation sequencing. As a conclusion, distinguish 3 sepsis pathogens from environment, contaminant, and the human commensalism is challenging, not only for molecular, also traditional blood culture. Sometimes when we found cons and also some fungi, it is not easy to rule out or to consider as a real underlying pathogen or not. So host microbes may facilitate the precision antimicrobial stewardship because it can discriminate sepsis from type of non-infectious febrile, can be inflammatory syndrome ranging from autoimmunity disease or macrophage activation syndromes. It can be also used at the time of emergency department presentation for all sepsis suspected patients. It is more accurate diagnosis to rule out sepsis compared to the available methods to diagnose sepsis. Acknowledgement, I would like to thank my home university, Arsi University, and also the funder, Federal Ministry of Research, Technology, and the Space, and also Charity, Truth, and the team organize this webinars lecture, and UKD, the whole STAIRS team. Thank you very much.
Tafese, thank you so much for your incredibly insightful presentation. I see hearts coming up on the screen because it was excellent. Thank you so much. First of all, I am going to look at the chat now and see if there's any questions from the audience. Please, everyone here, feel free to put your questions in the chat and or maybe raise your hand. I'm going to do my best and see who has their hands up. And if there's any questions, put them in the chat or please ask. I'm doing my best to read them as soon as I see them in the chat. In the meantime, let me, maybe I'm curious too. No, you see, there's Professor Emmanuel. Please, can we have the question from Professor Emmanuel? Sorry, I don't see your last name fully on my little screen here. So please just go ahead and ask what you want to ask.
Okay, thank you so much for. Thank you so much. Thank you. Yeah, I want to say a very big thank you to the presenter. Quite expository and engaging. My take on the presentation regarding the last comment in the conclusion session is that we have to exercise a little caution, especially in this era of antibiotics, antimicrobial and microbial resistance. to antibiotics usage. The point of a quick sampling of patients as at the point of presentation is very, very cardinal and can be management defining. Yes, we have to consider empirical use of antibiotics, but the point of quick sampling as at the point of presentation is very, very cardinal for culture, sensitivity, and as it may apply. So yes, low sensitivity, but culture is still gold standard, no doubt. But the point I'm trying to make is quick sampling of patients as at the point of presentation from all sites of possible sepsis focus is very, very important. Over to you. Thank you so much.
Okay, thank you, Professor Ma. Yes, the time of sample collection, especially for before initiation of antibiotics, matters the outcome of our lab activity or result. It is definitely true. This is not only for blood culture, by the way. It also affects the molecular, the cell-free DNA sequencing, because the life of this cell-free DNA is very short. If the patient already initiated antibiotics, there is high possibility that this bacteria already cleared, and the DNA also, as I already mentioned, it's about maximum up to 1 hour or 2 hours. If the bacteria is not there, there is no also cell-free DNA from this bacteria. So as early as possible, just collecting the at least blood for culture and also plasma and whole blood for further diagnosis. That is what I personally recommend. And also considering, based on the infection suspected and considering other clinical specimen also, I mean, can be the source infection. If it is peripheral, there is still, you miss from the blood culture, but the patient is still, sepsis, but you may miss it. So combination of can be sputum, can be urine, or can be any relevant clinical specimen. That also something recommended consider with the blood culture or collecting the blood for blood culture as well as for next generation sequencing. Or can be also targeted PCR, multiplex PCR.
Thank you, Tafese. This is fantastic. I see in the chat a big thank you also from Solomon Yingis to you. Thank you for the excellent updates. I found them very helpful. Me too, I have to agree. Is there any other questions from the audience? Please raise your hand, put anything in the chat that you always wanted to know about novel diagnostics. In the meantime, Tafese, let me just quickly, if I may, ask you, you like pointed out and made it very clear that this is these combination of methods can be extremely helpful at the time of first diagnosis to rule out sepsis, especially also. Do you think it has any, or is there any, I'm coming from a clinical side, I'm not, too much in these things, but do you think there's any value of the methods also, further down the course of the disease? So when the patient gets treatment and clinical course changes, maybe improves, is there any value of these diagnostics also down the path?
Yeah, as I said, the concentration of, for example, the cell-free DNA, it also tells us the progress of the patient.
Mm-hmm.
The half-life of shelf life of this cell-free DNA is very short. So if the patient is improving, the concentration immediately go down. That means you can associate. But if the cell-free DNA concentration is still increasing, it gives us, warning that the patient outcome will be bad. So we may change the management. And not only this, there are also other biomarkers which we also use.
It's the next generation of scientists is contributing as well. This is good.
So to consider other biomarkers also very important. Sometimes, this cell-free DNA, it also shows us, as I try to mention, if there is organ damage, that's also something helpful. Yeah.
Great. Okay. So it can be.
When we compare also with the blood culture, still, even if the patient initiated antibiotic, when we compare the positivity rate, it is better to focus to molecular than blood culture. The sensitivity is still better if we see cell-free DNA sequencing rather than blood culture.
Yes. Yeah, this is fantastic. This is very interesting and, a very novel path. I look at the chat and I see Uche from Nigeria with the questions. Thanks, Tafese, for the brilliant presentation. Did you say that NGS is not very good at detecting RNA viruses? If so, are there other options available? Because viral sepsis is a challenge in my part of the country, and I happen to know that this country is Nigeria.
Yeah. Thank you, Professor Uche, for a really nice question. Yes, when we focus to metagenomic next generation sequencing, even if we consider also RNA from plasma, RNA like DNA, cell-free RNA, we can do that, but it is not stable like DNA, and that also compared to that with cell-free DNA, if we compare cell-free RNA, it's not the same. It is better. The cell-free DNA is better. But if we consider sorry, could you mute yourself, please?
Exactly. Thank you.
If we really integrate with post-response gene expression and also the signature, that is it viral or bacteria, if it is viral, DNA or RNA, there are many things when the infection is there from the host. If we integrate with post-response, then we can definitely diagnose whatever, is it RNA, bacteria, or DNA virus. So if we only be focused to the cell-free RNA, yes, it is bad. It's not good. So the option is combining with host response.
No. This is, I think, an important takeaway in general, right? The combination of diagnostic methods in the future will give us like the whole picture of what's happening. Uche, thank you. Thank you for the question, Professor Uche. There's another question from Professor Emmanuel. Please go ahead.
Okay, thank you so much, Tafese, for the response to the question asked by Professor Uche. Well, I just have a few add-on to some of the comments that you had made. First, the way to look at the underpinnings for the modulations or the changes that you see in the interpretation of those results is as a result of the genetic component and the epigenetic component. You see, what is modulating the changes in picture that you are seeing is as a result of the genetic components from either those viruses, bacteria, etc., etc. Then the epigenetic factors, environmental insults, etc., can also modulate some of those pictures that you are seeing in terms of interpretation of the eventual results that you see. So in overall, just like Stephen just itemized now, multi-diagnostics is the actual way to go so as to guide management appropriately. Over to you, Tafese. Well done.
Yeah, thank you for nice question again, because when I say integrating with host response also, this is to increase the specificity, the sensitivity. Usually, we can use or we can cover by focusing to cell-free DNA or cell-free RNA, but for the specificity, if we particularly integrate this host response, that will helpful. In some cases, as you said, it is vary. Even, for example, among immunocompromised individuals, I don't expect the same host response, And also, some virus, they just to overcome the immunity of the host, they hide themselves. They know how to skip, the host response by down-regulating the response against to this, against to them. So there are still no definitive diagnosis, but when we focus to in general, if we integrate hosts and also molecular, next generation sequencing in metagenomic, then that will be better, particularly in case of sepsis, because there is also clinical, manifestation from the patient. Yeah, this is my answer, unless I know that it will be different from different hosts and then different pathogens. That is, you can't easily standardize it. But if you add like AI or machine learning, you can easily catch them.
Thank you, Tafese. We are coming close to the end of the hour. I'm still, if there's any burning questions still, we can make space for another one question. If there aren't any, then I'm just gonna, as you can see, there is many thank yous in the chat for the excellent presentation, very informative, very great update on what's new. And there's also from Ethan, as a fourth-year student nurse, this is actually very informative and quite alarming, because multi-drug resistant organisms can be quite hard to manage. Thank you so very much. Also from me, a big thank you. If there's no other questions at this point, then I thank everybody for joining this great session. It will be available also if you want to recommend or watch it again online. We have recorded the session and we will be uploading it online on our Sepsis webpage. Thank you so much for joining, and we hope to see everyone again in 2 weeks' time when we have our next session of the Global Sepsis Lecture Series. Tafese, to you again, thank you so much.
Okay, thank you very much all. Thank you, Petrol, for organizing. And I hope within STAIRS in the coming 4 or 5 years, we also have always some evidence. So, yeah. Thank you very much.
Yes, we will. Thank you so much. Goodbye, everyone.